Welcome to this lecture on the principle of immunology. Today, we are going to explore one of the most elegant and crucial concepts in biology, the clonal selection theory. If you have ever wondered how your body knows exactly how to fight off a virus it has never seen before, or why vaccines work so effectively, this is the theory that explains it all. Let us dive in and discover how your immune system achieves this incredible feat. To understand how our immune system works, let us start with a simple analogy. Imagine your body is a massive fortress, and an enemy spy, a virus, has just infiltrated your walls. Because you do not know what the spy looks like in advance, you cannot just build a custom trap on the spot. Instead, you already have billions of different pre-made traps scattered throughout the fortress. When the spy accidentally steps into the one trap that perfectly fits their footprint, that specific trap sends an alarm. You then immediately manufacture millions of exact copies of that successful trap. Your immune system does not design a new weapon from scratch, it selects a pre-existing one. Now that we have a conceptual picture, let us look at the actual biology. The clonal selection theory was first proposed by Frank McFarland Burnett in the 1950s. It is the fundamental principle that explains how our adaptive immune system achieves its incredible specificity. According to this theory, our bodies produce billions of unique lymphocytes, which are specialized white blood cells like B cells and T cells. The crucial point here is that these billions of unique cells are generated long before any infection ever occurs. They are the pre-made traps waiting patiently in your fortress. Building on the idea of pre-made traps, let us examine how these cells actually catch an invader. Each of these billions of lymphocytes expresses a unique, randomly generated receptor on its surface. Think of this receptor as a highly specific lock that can bind to only one specific molecular shape, which we call an antigen. When a foreign pathogen, like a bacterium, enters the body, its antigens will circulate and eventually bump into the specific lymphocyte whose receptor perfectly matches its shape. It is a game of molecular chance that results in a perfect, specific fit. So what happens when that perfect match finally occurs? This binding event is the selection in clonal selection theory. The antigen literally selects that specific lymphocyte out of billions. Once selected, the lymphocyte is triggered to activate and undergoes a process called clonal expansion. This means it begins to divide rapidly and exponentially. One cell becomes two, two become four, and so on, until a massive army of identical clones is created. Every single clone in this army possesses the exact same receptor, making them perfectly equipped to hunt down and destroy that specific pathogen. Before we move on to the mathematics of this expansion, we must highlight a crucial final step. After the infection is cleared, the selected lymphocytes do not just disappear. They leave behind long-living memory cells that patrol your body for decades. If that exact same pathogen ever enters your body again, these memory cells recognize it instantly and mount a massive defense before you even feel sick. This mechanism of selection and memory is absolutely critical because it forms the biological foundation for how vaccines protect us. A vaccine simply provides a harmless antigen to trigger this selection and build your memory cell army safely. Now that we understand the biological process, let us look at how immunologists model this rapid cellular division mathematically. Because one cell divides into two and two into four, this growth is exponential. We use a standard exponential growth equation to predict the immune response. In this formula, N of T represents the total number of cloned lymphocytes present at a given time. N0 is the initial number of selected cells, usually just one. The variable T stands for the total time passed, while T sub D is the doubling time, the time it takes for one cell to divide. As time increases, the exponent grows, causing the population to explode. Let us see this formula in action with a concrete example. Suppose a single naive B cell recognizes a flu antigen. It has a doubling time of 12 hours, and we want to find the total number of clones after 5 days. First, we must ensure our units match. We convert 5 days into hours by multiplying by 24, giving us 120 hours. Next, we substitute our known values into the equation. Our initial cell count is 1, total time is 120, and doubling time is 12. Dividing the exponent gives us exactly 10 division cycles. 2 raised to the power of 10 yields our final answer, 1,024 identical B cells ready to fight the virus. Now that we have calculated population size, this brings us to a more complex clinical question. In a severe infection, doctors need to know how long it will take to generate a massive response, say 1 million T cells. If the doubling time is 8 hours, how many days will this take? We start by setting our target population to 1 million, our initial count to 1, and our doubling time to 8. This sets up an equation where our unknown variable, time, is trapped up in the exponent. To solve this, we will need to use some algebraic rules to bring that variable down to a solvable position. To isolate a variable trapped in an exponent, we must take the logarithm of both sides. Using the base 10 logarithm is highly convenient here. On the left side, the logarithm of 1 million is simply 6, because 10 raised to the power of 6 is 1 million. On the right side, we apply the power rule of logarithms, which allows us to pull the entire fraction containing our time variable out to the front as a multiplier. We also know that the base 10 logarithm of 2 is approximately 0.301. We have now transformed an exponential problem into a simple linear equation. Let us complete the derivation to find our final timeline. We have our linear equation, and we need to isolate time. We multiply both sides by 8, which gives us 48. Then we divide that by 0.301. This yields approximately 159.47 hours. To make this clinically meaningful, we divide by 24 hours per day, resulting in roughly 6.6 days. This mathematical proof perfectly explains the biological reality. It takes nearly a full week to build a million-strong army, which is exactly why you often feel sick for several days before your immune system gains the upper hand. Having proven the timeline mathematically, let us visualize this entire elegant process. On the left of this diagram, you see the naive B cell pool. Notice how each cell has a uniquely shaped receptor: circles, squares, triangles, and stars. When the pathogen arrives, its antigens only fit the triangle receptor of B cell 3. This is the moment of selection. Following the large arrow, you see this single selected cell undergoing clonal expansion, multiplying into a massive cluster of identical clones. Finally, these clones differentiate into plasma cells that secrete antibodies to fight the current infection, and memory cells that will stay behind to protect you for the rest of your life. To wrap up our lecture, let us review the most important ideas of the clonal selection theory. First, remember that your body does not invent weapons on the fly. It relies on a vast, pre-existing library of randomly generated lymphocytes. Second, an infection is fought when a pathogen's antigen inadvertently selects its own destruction by binding to the one perfectly matching lymphocyte. Third, this single binding event triggers a massive, exponential cloning process to clear the pathogen. And finally, this process leaves behind a specialized army of memory cells, ensuring you are protected for the rest of your life. You now have a solid understanding of the fundamental principle of immunology.